Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to cover Chapter 3 for our MCAT Biology playlist. This chapter is titled Embryogenesis and Development, and here we're going to cover the following objectives. First, we're going to start by looking at the early developmental stages. That means we're going to go step by step through fertilization, cleavage, blastulation, gastrulation, and neurulation. Each of these processes marks a major milestone in the formation of the embryo. We're also going to take a moment to highlight what can go wrong during these early stages and how those issues can affect development. From there, we're going to move into the mechanisms that guide development. So we're going to talk about how cells become specialized, how they communicate with each other, and how they're guided to their proper locations in the body. We're also going to cover what happens when cells are programmed to die, how some tissues can regenerate, and what changes occur as the body begins to age. After that, we're going to take a close look at fetal circulation. This section focuses on how the fetus gets oxygen and nutrients before it's born, and how its circulatory system is uniquely adapted to bypass the lungs and liver during development. Finally, we're going to walk through gestation and birth. We'll talk about what's happening during each trimester, and we'll end this lecture by breaking down the process of labor and delivery. So with this introduction in mind, let's get started with Objective 1, early developmental stages. In this section, we're going to explore development from the moment a diploid zygote formed all the way through neurulation, which is the process that gives rise to the neural tube. This is the structure that will eventually become the central nervous system. Let's begin with fertilization. Fertilization occurs at the ampulla of the fallopian tube. This is the widest part of the tube. This is where a secondary oocyte encounters a sperm cell. If you remember from our previous chapter, a secondary oocyte has been ovulated, but it hasn't completed meiosis yet. For fertilization to happen, the sperm has to make its way through two protective layers that surround the egg. The corona radiata, which is the outer layer of follicular cells, and the zona pellucida. This is the glycoprotein-rich layer that's just outside of the egg's plasma membrane. The sperm then uses enzymes released from the acrosomal apparatus to break down these layers and make its way to the egg's surface. Once the sperm successfully penetrates the egg's plasma membrane, its pronucleus is going to enter the egg, and only then does the secondary oocyte complete meiosis II and form a mature ovum and a second polar body. At this point, the egg undergoes a cortical reaction, which is critical to ensure that fertilization only happens once. So in this reaction, the egg releases calcium ions, which causes the membrane to depolarize. This depolarization has two major effects. First, it prevents additional sperm from fertilizing the egg. This is a process known as polyspermy prevention. The second major effect is that it increases the metabolic rate of the newly formed zygote to prepare it for the next stages of development. The calcium release and the membrane changes also lead to the formation of what's called the fertilization membrane. This is an impermeable barrier that makes sure no more sperm can enter. Now that we've talked about fertilization, let's take a moment to discuss twins. Twins can form in one of two ways, either from one fertilized egg or from two separate ones. When two separate eggs are fertilized by two different sperm, we get dizygotic or fraternal twins. These twins are no more genetically similar than any other siblings, and each one is going to have its own placenta, chorion, and amnion. The placenta, by the way, is the organ that facilitates nutrient, gas, and waste exchange between the mother and the fetus. The chorion is the outermost fetal membrane that contributes to forming the placenta, and the amnion is the thin membrane that surrounds the embryo, and it fills with fluid to help cushion and protect it during development. When zygote splits into two embryos, then we get monozygotic or identical twins. These twins share the same exact genetic material because they come from a single fertilized egg. For monozygotic twins, the timing of the split really determines how many structures the twins share. And this has a lot of important clinical implications. Now, if the split happens pretty early on, meaning between 1 to 3 days after fertilization, the embryos are going to develop completely separately. That means each twin ends up with its own placenta, chorion, and amnion. This is called a dichorionic diamniotic pregnancy. If the split happens a little bit later, meaning between 4 to 8 days after fertilization, the twins may share the same placenta and chorion, but they're still going to have their own amniotic sacs. This is known as monochorionic diamniotic pregnancy. If the split happens pretty late, meaning between 8 to 13 days after fertilization, the twins are going to share everything. There's only one placenta, one chorion, and one amniotic sac. This is called a monochorionic monoamniotic pregnancy, and it comes with higher risks. If the split is incomplete, that means that the zygote doesn't divide fully, and we actually get conjoined twins, where the two embryos remain physically connected. And as a general rule, the more structures that are shared, the higher the risk for complications during pregnancy. That's why understanding the timing of the zygotic split is so important in prenatal care. Now, after fertilization, the newly formed zygote begins its journey through the fallopian tube towards the uterus, where it will eventually implant. But before it even gets there, something really important starts happening, and that is cleavage. Cleavage is the name for the rapid mitotic cell divisions the zygote undergoes during this early stage. And here's a key point. Even though the zygote is dividing, it's not actually growing in size yet. Each division just splits the cytoplasm of the original zygote into smaller and smaller cells. The very first cleavage is especially significant because it marks the transition from a zygote to an embryo. That first division is what officially starts embryonic development. Now, depending on how the cells divide, cleavage can be classified in two ways: indeterminate or determinate. Indeterminate cleavage means that each of the resulting cells still has the potential to become any cell in the organism. In other words, they're not locked into a specific role yet. This is the kind of cleavage that allows for the formation of identical twins because each cell is still capable of developing into a full organism. On the other hand, determinate cleavage means that the fate of each cell is already set. These cells are committed to becoming a specific part of the body, and they're no longer flexible in terms of what type of tissue they'll form. As cleavage continues, the embryo goes from 2 cells to 4 cells to 8 cells and so on, all while it's traveling through the fallopian tube towards the uterus. Once cleavage is complete, the next stage in early development is blastulation. After several rounds of cell division, the embryo becomes a solid ball of cells, and we call the structure the morula. At this point, the cells are still tightly packed together, and there's no visible internal cavity just yet. But very soon, fluid is going to begin to fill the inside of this ball of cells, and it will form a hollow structure called the blastula. The fluid-filled cavity inside is called the blastocele. In mammals, the blastula is more known specifically as a blastocyst. And this is where we start to see some specialization in the cells. The blastocyst is made up of 2 main parts. The outer layer of cells is called the trophoblast. These cells will go on to form structures that support the embryo, like the chorion and eventually the placenta. Inside the blastocyst, there's a group of cells called the inner cell mass. This cluster is what will eventually become the organism itself. Every tissue and organ in the developing body will eventually come from these cells. So at this point, the embryo has gone from a single cell to a structured, multi-layered sphere with both supportive and embryonic components. Now that the blastocyst has formed, the next step is implantation. The blastocyst continues traveling down the fallopian tube until it reaches the uterus. And once it's there, it's going to attach to the endometrium, which is the inner lining of the uterus. This is where the embryo is going to settle in and begin forming deeper connections with maternal tissue. Now, the outer layer of the blastocyst, which we called the trophoblast, these play a key role here. These trophoblast cells are going to start to invade the endometrium, and they're going to form structures called chorionic villi. These finger-like projections are going to help anchor the embryo, and they later become part of the placenta. The placenta is what connects the developing embryo to the uterine wall. And through this connection, the embryo receives oxygen and nutrients, and it's able to have waste products removed. The exchange happens through a structure called the umbilical cord, which links the embryo to the placenta. But early in development, the placenta isn't fully functional yet. So before it's ready to take over, the embryo relies on 4 structures for support. First is the yolk sac, which helps with early blood cell development. Then there's the allantois. This is involved in early fluid exchange. Next is the amnion. This is a membrane that surrounds the embryo, and it fills with fluid to cushion and protect it. Finally, the chorion surrounds everything, and it contributes to forming the placenta. So together, these structures keep the embryo alive and stable during the earliest stages of pregnancy until the placenta is fully developed, and then it can take over. Once implantation is complete and the blastocyst has settled into the uterine wall, the next major step in development is gastrulation. Gastrulation is the process that establishes the 3 primary germ layers of the embryo. It begins when certain cells on the surface of the blastula start to move inward. This movement is called invagination, and it transforms the blastula into a new structure called the gastrula. As cells migrate inward, they form a tube-like structure known as the archenterion, which will eventually develop into the gut. The opening to this structure is called the blastopore. And here we want to make an important distinction. In humans, which are deuterostomes, the blastopore develops into the anus. In contrast, in protostomes, the blastopore becomes the mouth. As this inward folding continues, some of the migrating cells also move into what remains of the blastocele, which is the fluid-filled cavity in the blastula. This movement establishes 3 distinct layers of cells. These are known as the primary germ layers, and each one gives rise to specific tissues and organs in the body. Let's go ahead and walk through each of these 3 layers. The first is the ectoderm. The ectoderm is the outermost layer. This gives rise to the skin, hair, and nails, but it also forms the nervous system, the lens of the eye, the inner ear, and the lining of the mouth. Any structure that interferes with the external environment or has a neural origin typically comes from the ectoderm. Next we have is the mesoderm. The mesoderm is the middle layer, and this one is responsible for forming the musculoskeletal system, including muscles and bones, as well as the circulatory system, the excretory system, and the gonads. Now, the adrenal cortex, which is part of the adrenal gland, also develops from the mesoderm. Finally, we have the endoderm, which is the innermost layer. The endoderm forms the epithelial linings of the digestive and respiratory tracts. It also gives rise to the pancreas, thyroid, bladder, and parts of the liver. Now, at this point, even though the basic body layers are set up, the cells themselves still need to specialize into their final forms. And this happens through selective transcription, which means only certain genes are expressed in each cell, depending on what it's meant to become. Another key concept here is induction. This is when one group of cells influences the fate of nearby cells through chemical signaling. The cells that receive these signals must be competent, meaning they're capable of responding. The molecules responsible for this signaling are called inducers, and they often guide nearby tissues towards specific developmental paths. Now, once the 3 germ layers have been established, the next major step in embryonic development is the formation of the nervous system. This process is called neurulation. The first structure to form is the notochord, which develops from mesodermal cells. Now, the notochord doesn't become part of the nervous system itself, but it does play a critical signaling role. The notochord sends out molecular signals to the ectoderm, which is the outermost germ layer, and it causes a specific region of it to thicken. This region is called the neural plate. As the signals continue, the edges of the neural plate are going to move inward and fold inward and essentially form what's known as the neural fold and the central neural groove. These folds continue moving towards each other until they meet and fuse and essentially form a sealed hollow structure that's called the neural tube. This tube will eventually give rise to the central nervous system, which includes both the brain and the spinal cord. Now, at the edges of this folding process, we also form a specialized population of cells called the neural crest. These cells break away and migrate to various parts of the embryo, where they're going to develop into a wide range of structures. This includes the peripheral nervous system, pigment cells, part of the adrenal medulla, and connective tissue in the head and neck. Now, one final note here, this stage of development is extremely sensitive to outside influences. Certain substances, known as teratogens, they can interfere with these early developmental processes. These include things like alcohol, drugs, certain infections, and even environmental toxins. Exposure to these during early pregnancy can cause some serious birth defects, or in some cases, even lead to the death of the embryo. With that, we've completed Objective 1, and now we can move into Objective 2, which is titled Mechanisms of Development. The human body contains around 37 trillion cells, and almost every one of them is specialized to perform a specific function. From neurons to red blood cells to skin cells, every type of tissue plays its own role, and each cell knows what to become thanks to a tightly controlled sequence of events. This process starts with specification. In this stage, a cell is reversibly designated to become a certain type. So it hasn't fully committed yet, it's more like a suggestion. That changes in the next step, which is called determination. Here, the cell becomes irreversibly committed to a specific fate. This decision is guided by chemical signals called morphogens, and the ability of a cell to respond to these signals is known as competency. After a cell has been determined, it begins to differentiate. Differentiation is the process by which a cell changes its physical structure and its internal machinery to carry out its specific function. So, for example, a cell that's destined to become a neuron is going to start building the proteins and the structures that are needed for transmitting nerve signals. Now, before differentiation begins, many of these cells actually start out as stem cells. Stem cells are undifferentiated cells with the potential to develop into a wide variety of other cell types. And depending on their level of flexibility, or what we call their potency, stem cells are classified into 3 main types. First, we have totipotent stem cells. These are the most flexible. They can become any cell type, including cells from the 3 germ layers and extra embryonic structures like the placenta. So a fertilized egg and the cells of the very early embryo, they fall into this category. Next are pluripotent cells. These still can form any cell from the ectoderm, mesoderm, or endoderm, but they can't make placental tissue. Finally, we have multipotent cells. These are the most restricted. They can only become a specific subset of cell types, usually within the same germ layer. Now, when cells begin differentiating, they don't just work in isolation. In order for tissues and organs to form correctly, cells need to communicate, and they do this through a variety of chemical signaling methods. The first type is autocrine signaling. In this case, the cell releases a signal that actually acts back on itself. It's like the cell is giving itself instructions, and this kind of feedback can really help reinforce a developmental decision or amplify a response. Next is juxtacrine signaling. This happens between two cells that are directly touching. This includes signaling through gap junctions where molecules pass directly from one cell to another. It's like a shared bridge for quick communication. Then we have paracrine signaling, which occurs between nearby cells. The signaling molecule is going to be released by one cell, and it's going to diffuse over a short distance to affect its neighbor. This is especially important in development because it allows cells in one area to influence the fate of adjacent regions. Finally, there's endocrine signaling, which involves long-distance communication. The signaling cell is going to release a hormone or a chemical into the bloodstream, and then it travels to reach a distant target cell somewhere else in the body. This is the most widespread form of signaling, and it plays a key role in coordinating changes across tissues. All of these signaling methods, they play a role in how cells respond to inducers. Inducers are molecules, often proteins, called growth factors that are secreted by one group of cells to influence another. These molecules trigger specific changes, and they usually promote differentiation and cell division in targeted tissues. Now, a common theme in development is reciprocal induction, where two tissues take turns influencing each other's fate. So, for example, one layer might signal the one next to it to start forming a specific structure, and that second layer then sends a signal back to refine the original. Now, this is so cool to think about because it's so similar to how humans get things done in like a group project or a research lab. We can also see the effect of morphogen gradients. Morphogens are signaling molecules that spread out from a source, and they form a concentration gradient. Cells that are close to the source receive high levels, while those that are further away receive lower levels. And depending on how much morphogen a cell is exposed to, it can activate different sets of genes and end up becoming different cell types. Of course, once cells have been told what to become, they need to get to the right place, and that brings us to cell migration. During development, cells often originate in one area, but they need to move to another location to do their job. Migration is guided by chemical signals, extracellular matrix proteins, and sometimes physical barriers or pathways. A great example is neural crest cells. These cells form at the border of the neural tube, but they don't just stay there. Instead, they migrate throughout the embryo and they differentiate into a wide variety of tissues. So when we talk about development, it's not just about the cell knowing what to become. It's also about knowing where to go, when to go, and who to talk to along the way. And that coordination is what makes complex multicellular life possible. And that is truly amazing. Next, I want to talk about what happens when cells die. That means we need to distinguish between apoptosis and necrosis. Apoptosis is a form of programmed cell death. It's highly organized and it's essential for proper development. During apoptosis, the cell shrinks, its DNA condenses, and it breaks apart into smaller fragments called apoptotic bodies. These are then cleared away by immune cells through a process called phagocytosis without causing inflammation. This helps sculpt tissues during development, and it eliminates damage or unnecessary cells. On the other hand, necrosis is uncontrolled cell death, and it's usually caused by injury or trauma. In necrosis, the cell swells, its contents begin to leak out, organelles break down, and inflammation follows. So it's messy, and unlike apoptosis, it often damages surrounding tissues. Now, another interesting thing that's worth discussing is regeneration. Regenerative capacity refers to an organism's ability to regrow parts of its body. Some organisms, like salamanders or starfish, can regenerate entire limbs. That's called complete regeneration, where the tissue is restored exactly as it was before. In humans, however, regeneration is typically incomplete. We can repair damage, but the new tissue may not fully match the original, and this, of course, varies by the tissue type. For example, liver cells can regrow after damage, but a severed limb won't regenerate. Finally, to wrap up this objective, let's talk about aging. As an organism gets older, both its cell and tissues undergo gradual changes. These changes affect everything from DNA stability to protein function to cell signaling. Over time, this leads to a disruption of metabolism. It leads to reduced repair ability, and ultimately it will lead to the death of the organism. At the cellular level, one of the key features of senescence is telomere shortening. Telomeres are protective caps at the ends of chromosomes, and with each round of cell division, the telomeres get shorter. Eventually, they become too short to protect the DNA, and the cell receives a signal to stop dividing. That's when cellular senescence begins. These cells remain alive, but they don't divide, and they contribute to aging and inflammation if they build up in tissues. With that, we've completed the second objective, and we can now move into the third objective, where we're going to talk about fetal circulation. This is the system that allows the developing fetus to receive oxygen and nutrients without using its own lungs or digestive organs. The key to this system is the placenta. The placenta is a specialized organ that allows the exchange of nutrients, gases, and waste between the mother and the fetus without mixing their blood directly. The mother's blood and the fetus's blood, they stay in separate compartments, and exchange only occurs across a thin barrier through diffusion. Because the oxygen pressure in the mother's blood is higher, oxygen naturally flows from the maternal blood into the fetal circulation. Similarly, waste products like carbon dioxide move in the opposite direction. They go from the fetus to the mother to be removed from her body. Now, one of the key adaptations that help the fetus capture oxygen efficiently is fetal hemoglobin, or HbF. Unlike adult hemoglobin, fetal hemoglobin has a higher affinity for oxygen, which allows it to pull oxygen more effectively across the placenta and then hold onto it longer. What this does is it ensures that fetal tissues get the oxygen they need, even though they're receiving it secondhand. Now let's talk about how blood actually moves between the fetus and the placenta. The umbilical arteries, they carry deoxygenated blood away from the fetus and towards the placenta. The umbilical vein carries oxygenated blood back from the placenta to the fetus. Now, it's the opposite of what we would usually expect, but that's how this system is wired. So remember, oxygenation occurs at the placenta, not in the fetal lungs. In addition to its role in nutrient exchange, the placenta also serves as an immune barrier and as an endocrine organ, so it produces hormones that support the pregnancy. Now, inside the fetus, there are 3 key shunts that reroute blood so it doesn't go through organs that aren't functional yet. The first is the foramen ovale. This is an opening between the right and left atria of the heart. It allows blood to bypass the fetal lungs, which aren't inflated yet. Instead of going to the right ventricle and out to the lungs, blood flows directly from the right atrium to the left atrium and then out to the body. The second shunt is the ductus arteriosus. This vessel connects the pulmonary artery to the aorta, which again allows blood to skip over the lungs. So blood that does enter the pulmonary artery is rerouted into systematic circulation. The third and final shunt is the ductus venosus. This one allows oxygenated blood from the umbilical vein to bypass the liver and go straight into the inferior vena cava, where it can then enter the heart and be pumped throughout the fetus. So together, these three shunts ensure that blood is prioritized for essential organs like the brain and heart, while skipping over the lungs and liver, which will only become fully active after birth. And with that, we can move into our last and final objective, which is titled Gestation and Birth. Human gestation lasts approximately 280 days, or about 40 weeks, and it's divided into 3 trimesters. Now, a good general rule to remember is that the larger the species, the longer the gestational period, and the fewer offsprings that are produced per pregnancy. Now let's walk through each trimester and highlight the major events. In the first trimester, the most important developmental process is organogenesis. This is when the major organs begin to form. By the end of the first trimester, most of the embryo's basic structures are in place, and it's now considered a fetus. The second trimester is marked by rapid growth. The fetus starts to move, the face becomes more human-like, and features such as fingers and toes become more distinct. This is the stage when pregnant individuals often begin to feel movement, sometimes referred to as quickening. In the third trimester, growth continues and brain development accelerates. Another major event in this trimester is the transfer of antibodies from the mother to the fetus through the placenta. And this passive immunity really helps protect the baby after birth while its own immune system is still developing. Finally, let's talk about birth. Birth begins when the cervix softens and thins out. This is a process known as cervical effacement. Then the amniotic sac ruptures, and this is commonly known as when a person's water breaks. This is then followed by a wave of uterine contractions that begin, and they're coordinated by 2 key hormones, prostaglandins and oxytocin. These contractions push the fetus downward and eventually result in delivery. After the baby is born, there's one more stage, and that is that the placenta and umbilical cord are expelled from the uterus. And this final step is known as the afterbirth. With that, we've completed the journey from fertilization to delivery. I hope this helped. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.